Hello, everyone. Welcome to our webinar on soil health, the Rhizophagy cycle, and the Haney Test. My name is Youwakim, and today I'm joined by Dr. Liz Haney and Dr. James White. We are going to talk about a little bit what we do at Biome Makers, as well as the Rhizophagy cycle and how you can incorporate different methods of soil testing to make more informed on-farm management decisions. As we go through the webinar, feel free to write your questions in the chat. We will save the last 15 minutes of the call for questions from the audience. And we'll talk about plant microbe interactions, the rhizophagy cycle, and these different methods of testing. So welcome onto the webinar. Feel free to take notes if you want to save your questions to the end, but also know that if you put them in the chat, we'll get to them hopefully before the end of the call. So for those of you that are not familiar with BCRUP technology, here at Biome Makers, we are DNA sequencing soil. We're working with a database of 24 million microorganisms. So it's really exciting. It's a really large soil database. We are doing, after the DNA sequencing, we're running the samples through this database and providing agronomic insights based on that, as well as providing data over 36 different categories. We have 34 peer-reviewed publications, and we test soil from all over the world. So thanks to all of you for participating in that and sending us your soil samples. We know that soil is alive. This is really, new science that we're working with. We're working within soil as the system that it is to improve food quality. We're looking at biodiversity, functionality, resilience of soil to inform soil health, and the different, using microbes as bioindicators. So we're looking at all these different indices, thinking about soil quality, plant health, nutrient cycling. We are identifying arbuscular nectomycorrhizal fungi, looking at their ratios and how that informs soil health, detecting soil-borne diseases, looking at their biocontrol agents and different phytohormone producers, all of which help inform crop health and management decisions on farm, as well as when we're considering applying different soil amendments as needed. And of course, as we're very familiar with soil testing, when we look at nitrogen, phosphorus, and potassium, we're doing the same in biology. We're detecting microbes that are cycling nitrogen, phosphorus, and potassium, as well as carbon, and facilitating organic matter breakdown in different micronutrient pathways, as well as looking at different chemistry analyses to pair, so that we're looking at soil health in this holistic way. So besides that, the next step of thinking, besides what's in soil, we know that 1 teaspoon of soil contains billions of microorganisms, and sometimes people hear the word microbe and they immediately think, harmful or pathogenic, but we know that many microbes are extremely beneficial. And if we promote those beneficial soil microbial populations, then we are, let me see, what just happened? There we go. We are helping to increase yield, crop resilience, and nutritional quality of produce. So different plant microbe interactions, when we're looking in soil and let's say we detect a pathogen, but we can also detect different biocontrol agents. So different bacteria or fungi that are able to help defend against disease. That's really helpful to know and to help us analyze if we are applying the right amount of different biostimulants or different soil amendments that can help us with this biocontrol defense. And when we're looking at mycorrhizae and their ability to increase the capacity for roots to uptake nutrients, whether that's through length in the soil or reaching different nutrients that they otherwise wouldn't be able to reach. So that's colonization that we like to see. And there are a lot of different ways to look at that, whether you're DNA sequencing soil like we do in a biological way, thinking about different chemistry tests like Malic, which is widely known, physical chemistry tests like soil texture and bulk density. And today we'll talk a little bit about the Haney test with Dr. Liz Haney, who's on the call with us, and will give us more insight into what the test covers and how it covers both biology and chemistry. Liz, if you want to jump in here, I can go over this slide.
Oh, I can do it. Sure.
Okay.
The Haney test is quite a bit different than the standard test. We use different extractants. So she has a phosphorus up here. We use the H3A extract to look at phosphorus levels, plant available in the soil. H3A is comprised of malic, citric, and oxalic acid. So it mimics plant roots. It is not buffered, which means that when we add it to a soil, it stays within 1 pH of the actual soil pH. This is important because malic 3 is buffered to a pH 2.5. Bray is buffered as well. So when we add these to soils, it alters the soil condition. And what we often find, like with malic, for example, if you have high levels of calcium phosphates in your soils or iron and aluminum phosphates, we can tend to overestimate the amount of phosphorus that's available using the standard extracts. So we want to try and mimic what the plant actually sees in the soil by exuding plant root exudates. We also use water to extract nitrogen, which is a novel idea because it rains water. And that's what the microbes see, is what becomes soluble with water when it rains. And the plants also get to see that nitrogen that's available. We measure organic nitrogen and inorganic nitrogen. And then for all of the micronutrients, we also use H3A. So again, the basis for all these extracts is that we're trying to mimic what the soil sees in the field, in your field, as best as we possibly can without altering the whole entire soil chemistry using these harsh extracts. I don't know if there's anything else you want to add to that.
No, that sounds great. We have another slide I'll go to.
Okay. Yeah, so the Haney test was developed for fertility recommendations. My husband, Rick Haney, developed it. He was a farmhand for the majority of his life, and he kept seeing that farmers, one, as everybody knows, or most people know, producers don't have control over commodity prices or input prices, or for sure the weather. So Rick went back to school and tried to develop more robust fertility recommendations so that growers could get the most bang for their buck out of soil testing and the fertilizer that they're applying. But what it also looks at is the dynamic carbon portions in the soil. So most tests, when you send it to a lab, will look at total soil organic matter. That's the big house of soil organic matter that's in your soil. We also look at water extractable organic carbon, and that is the carbon that the microbes see. So those are shorter chain carbons. They become water soluble, the microbes can chew on them and cycle nutrients, which will become available to the plant. So for every piece of carbon in the soil, there's also nitrogen and phosphorus and other minerals attached to that carbon particle. Then we also measure the CO2 respiration from the soil, which is correlated with microbial activity and nitrogen mineralization. Only about 1 out of 10 labs in the country test for nitrate nitrogen. Usually it's just to the west of the Mississippi River. But we measure for nitrate, ammonium, and organic nitrogen. Research has shown that plants actually prefer to take up organic nitrogen over inorganic nitrogen because they can use it more efficiently in their systems. But We've also found that organic nitrogen accounts for, on average, 30 pounds of additional nitrogen that we weren't accounting for before. So depending on fertilizer prices, let's say they're $1 a pound for nitrogen, you can right off the top save about $30 in your fertility budget just by running this test, an acre. We look at organic nitrogen to inorganic nitrogen ratios. Again, organic nitrogen is favored by the plant, but it's also less subject to being lost by environmental conditions, so leaching or volatilization. We also look at organic phosphorus, which is something that most laboratories don't look at. Again, this is available to the plants. Microbes can chew on that organic phosphorus and make it available to the plants. Like standard tests, we also do look at potassium, calcium, all the micros. The Haney test does not measure for boron. If you want boron run with a Haney test, we recommend you do hot water soluble boron. And this is mostly because boron is a component of the glassware that we use to measure soils with and extract them in the laboratory. And so we don't want to get false numbers using that glassware. So we run it using a different test. And then like standard tests, look at soluble salts, pH, buffer pH, carbon and nitrogen ratios. Our carbon and nitrogen ratios that we look at are not the total carbon in the soil, that big soil organic matter house, and the total nitrogen. We look at that active portion, so the organic carbon, the organic nitrogen that's water soluble, and look at those ratios because that's what the microbes are responding to in the soil. And we want to see if those are in balance so that you're most efficient. We look at microbial respiration, so your biological activity, and the percent MAC, which is basically how active your microbes are relative to the amount of their food source that they have in the soil. And we can tell all sorts of information from this, which if people have questions about that, we can go into that later. Standard tests don't look at this portion. Again, we do fertility and Lyme Rex, just like standard tests, but we can also help with cover crop recommendations based on that water soluble organic carbon and nitrogen ratio. We can tell a lot about the way the soil's functioning by how active the microbes are relative to their food source, so that percent MAC. We can pretty much get a good idea of whether your soil's well aggregated, whether you have enough water infiltration and airspace, because if your soil is completely compacted, your microbial respiration is going to be low. So we're able to make practice recommendations as well based on these results.
Excellent. Let's see, got one more. So when we are, I think Dr. White, this is an excellent time to share a little bit about the work you do, and then we can talk about soil organic matter and how this all relates.
Okay, I'm happy to talk about the work that we do. We're at Rutgers University, and we study plant microbes and soil microbes. A lot of the soil microbes actually become plant microbes, and what happens is plants will these microbes will grow in the soil, and they will actually feed on organic material in the soil, and they'll establish symbiotic interactions. Bacteria will establish symbiotic interactions with fungi in the soil, and you'll have other microbes in the soil that like algae that grow in the system. Basically the food is the organic carbon that these microbes are using, but plants will attract these microbes to their roots through their secretions from the root tips of exudates, and those exudates are sugars and various forms of carbon oligosaccharides, organic acids. Sometimes it's vitamins in the exudates and other kinds of nutrients. Those are actually attractants to the soil microbes. Microbes come to the root tips where they're absorbed into the roots and they become endophytes. Endophytes is a really internal phyte plant. These are microbes that go into plants, or I should say it another way. These are microbes that plants absorb into their roots and cells. And some of those endophytes will actually go up into the seeds and fruits and leaves and so forth. But the plant in the roots will absorb the microbes into their root tips, into their cells, and then extract nutrients from them using reactive oxygen, superoxide, and then get nutrients from the microbes internally in their cells. And this is important. This process is happening in the cells of the plant. And the process that I'm describing is something that's come to be known as the rhizophagy cycle. Rhizo, root, phagy, eating, root eating cycle. This is basically where plants will absorb microbes into their cells and get nutrients out of them using reactive oxygen. And it's a cycle because the plant will cultivate, once extracted nutrients from the microbes in the cells, then the plant will cultivate those microbes and replicate them and then put them back out into the soil in root hairs, and so they will, when these root hairs are growing, they will basically eject microbes back into the soil. So you have microbes coming in at the root tip, microbes going out at the root hair tip. And this is a cycle, this rhizophagy cycle happens in all plants at some stage, usually in the early stage, but in the growing root tips, this is where rhizophagy cycle is happening in all growing root tips. But endophytes are all over the plant, and they're helping plants. And all of these, and I should say, so when you think about the microbiology that's important in growing crops in biological agriculture, right? What is the microbiology that's important? It's the soil microbiology and it's the plant microbiology. And both of those work together to ensure that you have good, healthy plants. And so it's both sources of microbes are important. I think that's the Joy. I think I covered the high points there. Did you want me to say that? Is that okay?
Yes, great starting point.
Okay.
I have some questions for both of you that we hear often, and we can let the discussion go as it does. First, for Liz, Dr. Haney, can you speak to farmers' hesitations about using a newer soil chemistry test when they're familiar with other tests, and how the Haney test uses, without this standard calibration that we have for the other tests, how the Haney test is able to, once you have a baseline, how you're able to move forward from it?
Sure. I guess the biggest thing I hear from producers is, as far as using the Haney test, is that a lot of them don't understand what it means when they get it. So they're used to looking at their university recommendations, or they're just more familiar with it. So a big part in getting producers comfortable with using the Haney test is education, which we do a lot of. But also there's quite a bit of maybe negative feedback sometimes from certain people or universities or whoever that say our test is not calibrated. So the way that standard soil tests are calibrated is that there are research plots. Let's say they're growing corn. They put out a certain amount of nutrients until those nutrients become where they're not limiting for plant growth anymore. Take the yield and do soil tests and figure out what that yield response was to fertilizer applied. And that's how soil, and then they relate that to the soil tests. So it's yield, fertility applied on some certain soil under a conventional management system, usually done in different places throughout the country, but not everywhere. And so that's how it's calibrated. What the Haney test does is uses, to our best available knowledge right now, what actually happens in the soil to see what's available to your plant. So plant root exudates and water. So we are calibrated to your field under your conditions, under your climate, because we're measuring your soil and what it's going to see when water hits it, or when the plant roots are trying to dissolve nutrients and take them up. And also when the plants are feeding the microbes and how active they get in their nutrient cycling and their activity. Biology, soil biology is very, very complex, and we don't have all the answers, but this is the best that we have available right now, to my knowledge. And there are other tests that are also beneficial, where we can see what's happening with the soil biology and nutrient cycling in your active system, under your climate and your context, and not just what happens in the laboratory. I hope that covered it, but you can
Yes, no, great.
tell me to expand if you want me to.
Very thorough answer. Let's see. Dr. White, do you have any, or do you have an example of any clear differences you've seen in your work between different types of plant tissue? Any insights that you have been able to differentiate between different types of plants, or different variables that you were studying?
Well, I could tell you, I'll make a couple of statements. May not be exactly what you asked, but I'm going to address what we're talking about. These tests that we're coming up with are really needed, you know, like the Haney test and the bee crop that Biome Makers has developed, because tests in the past have focused exclusively on the chemistry. And that we know that that's only partially correct, partially gives us information, and it really gives us very little information if you're not actually fertilizing with chemistry, right? If you're doing regenerative agriculture or organic agriculture, these soil tests are really skewed. They're not giving the information that the growers really need, you know, that relate to the microbiology in the soil. And so this is really important, and we're really only now on the ground floor, to be honest, on developing these tests. There's a lot that needs to be done to make this kind of testing for biological agriculture improvements. And those improvements, I know they're on the way. I know they're being these tests are being continually improved. So that's happening. In terms of how plants respond differently, Joy, I'm not sure exactly what you mean by that, but I'll say some plants are, for example, trees are highly mycorrhizal, right? And those plants, they'll have, oftentimes they'll create ectomycorrhizal mantle that'll go around the roots. And of course, those roots are pretty much the nutrients going into them are pretty much being controlled by the fungus themselves, right? The fungi that go out into the soil. So that's very different than, say, for example, a grass plant that grows in the soil, in which the roots are highly branched with a lot of root tips. The grasses are doing rhizophagy cycle a lot. Whereas those trees with the mantle, with all the big roots, with all the big mycelium all over the root, they are leaning more towards this other kind of biology, right? These ectomycorrhizal associations. And, you know, so those are two key differences. I'll say also some plants, and I'm going to cheat a little bit here. Consider mushrooms, right? Mushrooms, I'm cheating a little bit, going back to the old concept of plant, mushrooms. They actually will do instead of something like rhizophagy cycle, they will absorb, they will get their own nutrients, doing their own little physiological thing to get nutrients. And then they will have inside them, they'll have bacteria in their hyphae and in their fruiting bodies. Inside the hyphae of the fruiting bodies, they'll have bacteria that fix nitrogen. And so those fungi will actually get the nitrogen, will get nitrogen directly out of those bacteria that are inside their hyphae. Okay, so you have and you go back, if you go back to the very earliest plants, the mosses, they work more like the fungi do than our modern crop plants, right? They don't do rhizophagy, but they get microbes in where that fix nitrogen, and they get the nitrogen out of those microbes. Whereas most of our plants are doing a rhizophagy cycle, and they're putting bacteria that fix nitrogen all throughout the tissues of the plant, and they get little tiny bits of nitrogen from those bacteria in the plant tissues. So, and of course, all those go back to soil microbes. So the importance of that microbiology in the soil. Did that kind of address that, Joy? It was a lot. Yes. And kind of rambly, but.
And I guess
excellent.
Besides one of his, if you don't mind,
go for it.
We haven't identified hardly any of the actual microbes that are in the soil. We don't even know what they are, but we got to start somewhere, right? And we do know what some do and what they function as. So yes, we are continually learning more, but it takes everybody doing all of these different things. Like we have done soil testing on certain things that we may not have seen the results through respiration or an increase in available nutrients right away. And that's because then we send these things to Dr. White, and he finds out they're doing completely different things within the plant tissue that we wouldn't have been aware of before. So there's definite advances and more that can be made after that.
We really are on the ground floor of understanding how to grow plants with biology. And it is exactly what Liz said. You know, it's going to take all of us, because we frankly, most of our concepts for agriculture come from this chemical agriculture mentality and chemistry, the way that works. And of course it works, but it's not actually how plants grow. It's not actually how plants grow. So all of the stuff that we're trying to figure out about, you know, how to maintain good soil microbiology, and how plants are using those microbes once they absorb them, how they're signaling to the microbes in the soil. All of that is just kind of cursory. You know, we just see a little bit of that, and you know, we've got to, and that's one reason why I personally always am inviting people to work on these soil and plant microbes and how that works in plants, because we need more people working on it. We need more. It's going to take a lot more than just a few of us who are out there toiling away trying to develop tests or develop understandings. It's going to take more.
Yeah, and I don't know about you, but when I went to school, I was taught under that chemical reductionist mentality. And so back to your question earlier, Joy, of, you know, why a farmer might not want to use the Haney test or learn about all these complex interactions is, one, it's a holistic way of thinking that none of us were really trained to do in the first place. It's just weirdos like me and James and, you know, my husband, our whole group. But it's hard to think about all these different interactions, and it can get overwhelming. But that's why we all work together holistically. to figure all this stuff out.
That's why I like to go to meetings, like the Acres meetings and there's the meeting Beyond Yield that you helped organize. Those kinds of meetings, people add a lot to understanding. People are very good, especially people in this biological agriculture, regenerative agriculture area, at making observations. And they come to get some real insights. So I've learned a lot going to these meetings, and I pay careful attention. What we oftentimes get wrong is the mechanism for why something works a certain way, but we often get right that it does work that way, yet not the details of it, right? Not the specific mechanism. So that's questionable, and in time, we'll learn about why actually the mechanism of why something works a certain way. But the first observations that something is working a certain way is that you can gather at these meetings. A lot of observations like that, and that's again the intelligence of a group of people who are trying to do the same thing and thinking about it, and it's really impressive to me.
Yeah.
About how smart people are.
Well, another thing I've really learned in the past few years, especially hosting these meetings and working with all the producers, is that the farmers are the real scientists on the field. And regenerative farmers especially are totally willing to experiment and tell you where they messed up and what happened and when they applied something, and then we can go and test it in the soil or see what the plant interactions are. And it's not just an academic exercise. It's all these different people working together, sharing information so that we can all figure stuff out. I don't farm. I don't know how to farm. I know things about it, and I know the science behind it, and I'm learning more every day, but it takes like farmers educating me on different things, and then us conversing together to really figure things out.
And they're brave too. They're brave, Liz, too. They're very courageous, doing their experiments and coming up with their own ideas, creative and brave, and radical too. I mean, this is the other thing, is they tend to be not just taking people's, if they're extension or whoever it is, don't take people's word for it. They really are quite progressive in that way. Like you said, there's scientists in their field. Yeah.
Dr. Haney, do you think, when you're talking about regenerative farmers, can you give us an example of how the Haney test has been used to inform regenerative practices?
Sure. Well, I'll give an example of my business partner, Russell. So he doesn't have a college degree, was a firefighter, career firefighter, and then a first generation farmer. And because he didn't have maybe his grandfather and great grandfather telling him this is how we did it, he had to go out and learn and teach himself and watch YouTube videos. And he was brave enough to call everybody and read papers and papers and papers and educate himself. And he found came to see us about 10 years ago, and other scientists, and started using the soil testing to monitor and track his progress on the regenerative path. And so in particular, he's been able to see where a certain practice has improved soil biology. And this is a lot of producers. They'll test the soil in the spring, apply fertility. Some of them test again mid-season through the fall, depending on how curious they are. And over time, they have the management practices. They know what they've been doing. They're willing to write them down and share them. And then we have been comparing that for many, many years now to what the soil tests look like, or the Haney test looks like. So has biology improved? Has microbial activity improved? And we get confirmation that these practices are improving nutrient cycling. In addition, they're reducing their fertility inputs, in especially nitrogen, and seeing the improvements in different, like maybe biological products that they're applying, or manures, or whatever it might be, based on the Haney test results. So it gives us information for fertility ahead of time. It gives us confirmation that our practices are working after the fact, or during the farming season. And they're all willing to share these results with each other, and we talk about it at all these conferences that Dr. White was mentioning. And so other people can then learn from what the producers are doing and how it looks on a soil test, so that we have a larger database of information to help other growers.
Excellent. So the Haney test, with the oxalic, malic, and citric acid, it's mimicking the most common plant root exudates. So that's really helpful. We're trying to see what how the soil is responding after rainfall, and what's realistic. Dr. White, can you answer what can a farmer do to promote the rhizophagy cycle and increase the population of endophytes?
That's a good question. Well, my answer is really simple and but multi-part. One is to build your soil, build the soil organic matter. Build soil, make soil, start to grow your soils. So use cover crops, build organic material, use compost if you're gardening, if you can. But otherwise, you build your soil, use cover crops, and leave some of the material in the ground. Leave most of the material in the ground. So you build your organic material, you build your soil carbon, and what you're going to be doing is to be increasing the life in the soil, the microbial life. And you can see soils are going to be living. They're going to have evidence of life in them. Worms, for example, will be in there. You'll have good organic material. The other thing is the cover crops are really living cover crops are very important, because they actually donate microbes to the soils and to your crop that grows there in those soils. And every plant has different microbes. So the more cover crops you use, the more endophytes, or microbes, bacteria, and fungi come out of those cover crops to increase the diversity of microbes that the plant can get. So cover crops is a way. Also, there are certain practices that we use that actually they're very ancient, but the biostimulant microbes, people use biostimulant microbes, things like bacillus and any bacterium, the ferments that people use, the compost washes that people use. Those are full of microbes and nutrients, but full of microbes. And those microbes, then when you put them on the soil around your plant, the plants can get those microbes. And so people do that. They will apply microbes directly to soils and to plants, and then you can put microbes as either around the roots, or you could put them actually as a foliar, and they'll go in, and that's because the microbes colonize the meristematic tissues of the plant and go into those tissues. And you have that in the shoots, the shoot meristem, and you have it in the roots, the root meristems. And the plants take them in both ways. Of course, if they're in the shoots, they can't cycle them, but if they're in the roots, they can cycle them. If you put them on the shoots, they can actually mobilize them down to the roots too. So it goes actually both ways inside the plant. What else? Those are some key ways. Of course, anything that adds microbes to soils is going to be a positive. So animals, for example, if you put animals, cows, or putting down manure, that'll add microbes to the soil. It should be composted though, it shouldn't be raw manure of any, because those are going to be too many microbes and that will overwhelm plants if it's not composted. So those are some things that can be done to, and the plants will just automatically get those microbes. It's not gonna be hard. If you get the microbes there, the plants will attract certain ones, and they don't get all of them, but they get certain ones, and those are the ones that function in the rhizophagy cycle. So I think, how was that, Joy? Did that address it enough?
Excellent. Yes, excellent. Thank you so much. Very thorough. So now we are going to get into our questions, and I'll summarize our discussion here for everyone. I know I was watching some people hop on later, but essentially all this work, the bee crop testing, Dr. White's studies on the rhizophagy cycle, and the Haney test from Drs. Haney serves to allow land stewards and farmers to make more informed decisions about how to be working with the soil system in a more synergistic way. So asking different questions about soil amendments or cover crops or different management practices, incorporating this testing will help to enhance yield and serve for more successful growing seasons. So thank you both for sharing about your work, and I will start asking you the questions we have from the audience here. Dr. White, I also have your slides here. If you want to refer to any of them, let me know. They're all here at the end as we answer these questions. So let's see, we do have a lot. I definitely won't be able to get through all of them in these 13 minutes, but we can have them emailed out to everyone who attended the webinar. So let's see. Dr. Haney, do you also evaluate nitrification and denitrification rate of soils?
We don't evaluate the rates of those, no, not directly.
Do you also calculate DOC, dissolved organic carbon?
Do we measure dissolved organic carbon?
Yes.
Water extractable organic carbon, which would include dissolved, and then CO2 respiration, we measure that. We don't fractionate carbon portions except for soil organic matter, water extractable, and the gas phase. So we're measuring the solid, liquid, and gas phases of carbon in the soil.
Okay, excellent. For Dr. White, what are the nutrients that are important to encourage the rhizophagy cycle, and are there certain conditions that are antagonistic to the rhizophagy cycle?
That's a good question. Yes. If water, for example, is contaminated with sodium bicarbonate, if it has bicarb in the water, right? Then the rhizophagy cycle would not function very well. The microbiological effect for some reason doesn't work, and the explanation likely is that the bicarb binds up the metals, and so the microbes can't get the metals, and they can't bring them to the divalent metals, bring them to the plant. So that kind of messes up the system. The overuse, I would say, of chelating herbicides or any chelating chemistry can cause problems. And of course, part of the problem is that the microbes need those metals as well, need those things like iron and manganese and well, those kind of divalent metals, magnesium, zinc. If the plant can't get them because these chemicals like herbicides that chelate bind them up, then the plant can't do rhizophagy cycle, and they can't get nutrients from those microbes. If the soils are flooded, then plants actually need oxygen in the roots in order to produce the superoxide to extract nutrients from the microbes that they internalize. And so if they can't get those nutrients, if they can't get those, if they can't get oxygen, then they can't make superoxide and they can't extract nutrients from the bacteria. And then plants that grow like that in flooded soils will actually go high in ethylene. And part of that is because that oxygen is not there to deactivate that ethylene. Normally superoxide would combine to ethylene and it would go. So then the stress actually builds up in plants. So those are some things that can happen that would interfere with rhizophagy. And of course, adding microbes and organic material, looking at soil health, that would increase rhizophagy. So those are just some things that happen that affect rhizophagy cycle.
All right, thank you. Dr. Haney, if a soil sample is done at V6 in corn, can the nitrate nitrogen results be used in determining a ballpark figure for the amount of side dress nitrogen needed?
Yes. But also do tissue testing if you can.
Okay, excellent. If the intention is to replicate arbuscular mycorrhizal fungi, what is the best plant phase to collect from the rhizosphere inoculated? And with microscope information.
It sounds like a question for me.
Yes.
Okay, sounds like a question for me.
I'm glad.
Yeah, obviously seedling stage is too early, right? Because the way VA mycorrhizae work, and I mostly, in our cultivated fields, we're talking about remnants of VA, vesicular arbuscular mycorrhizae. Those mycorrhizae don't, they normally will sit in the soil as a chlamydospore or a big spore. And when the plant root grows near it and sends out exudates, attracting microbes, those spores will germinate, and then they'll colonize the root externally and internally. So a bit older, sampling the roots when they're a little bit older. Also the soils, the best soils for any mycorrhizae are going to be good microbial rich soils that are not tilled extensively, right? Because when tillage, you move the mycorrhizae and all the bacteria, especially if you're doing any deep turning, you damage them. And by tilling and leaving the soil there open like that, a lot of those microbes are going to die, including the mycorrhizae. So the healthy soils and plants that are a little bit further along in development, not looking at the very earliest seedling, giving the plant a chance to get colonized, giving the mycorrhizae a chance to go on to the plant roots. So that's my answer there.
Great. Dr. Haney, is the Haney test available in Mexico?
There aren't any laboratories in Mexico that run it right now that I'm aware of, but you can contact Regen Ag Lab and get a customs form and send samples from Mexico to their lab or anywhere in the world.
And it's $55, correct?
Correct.
Okay. If we are to change the urban growth of urban agriculture, how do we develop a deeper understanding of using the test to determine the health of society? Well, I can give a summary answer to this, and then if either of you want to expand, that is good. If we're looking at urban agriculture and all agriculture, using these tests will help us increase nutrient use efficiency in soil, and then also with plant tissue. So we'll be growing crops that have higher nutrient density. And then when we're talking about health of society, soil really is the foundation of our public health. So the more that we're taking care of it and stewarding it, the higher quality produce we have. And they say you are what you eat. So we're eating higher quality produce, and our bodies have a better chance of protecting us against infection and having overall health. If either of you want to add to that, feel free.
Well, you can look up also David Montgomery's work on comparing the nutrient density of foods grown organically, conventionally, and regeneratively. And they did some studies in vegetables, meat, grains, and crops or livestock that's grown on regenerative soils, which have more healthy microbial populations, better aeration, water infiltration to get that oxygen down to the root, like Dr. White was saying that the plants need to take up the nutrients, you know, and to function, that those plants are healthier and that the grains are healthier. And so that's just, there is documented evidence, and we're doing more and more testing on that all over as well.
I could add something a little bit to that. So biological crops grown, foods grown with lots of microbiology are probiotic. They build the microbes in our guts, and especially when we eat them, eating vegetables and so forth, and crops that are not cooked too much, foods that are not cooked too much, will build our own gut microbiomes. And of course, that equates to health as well. In fact, one of the big problems we have in our society is that the microbiomes are collapsing. And a lot of that is because we're not having microbiological rich foods. And so we're not taking in new microbes. When you compare to people who live in the jungle and eat wild plants, their microbiomes are very diverse. Ours are very narrow, and we're getting all kinds of diseases from it. This actually is the, thank you, Joy, for putting this up. This is the cycle. This is the rhizophagy cycle that I was talking about, where plants are taking up these microbes. And you can see the cyclic nature. The microbes are attracted to the root tips. They're absorbed inside the root cells. So they're actually in the root cells. They are using superoxide to extract nutrients. The bacteria go into the root hair initials, and then they come out of the root hair tips. They actually trigger growth of those root hairs. Without the bacteria, you get no root hairs at all. But if you look at the little points there of the hairs, those are bacteria that are being ejected out of those root hair tips. They're coming out of the roots. So plants are getting those microbes and getting nutrients out of them, and then replicating them and putting them back into the soil. So they're cycling that. That's the rhizophagy cycle. They go like that around and around from the soil and back, and the plants increase the ones that work with it. So, but back to the microbiome part, we need healthy microbial rich foods for our own health too, for our own microbiomes. So there you go.
Thank you. Dr. Haney, who gives an interpretation of the Haney test for the grower?
Me. There are other people that do it as well, but yeah, we spend a significant amount of time on that. So if you're interested, I would tell you to email me right now, but my domain is down. So email me tomorrow when I have it fixed, and let me know if you need help with interpretation.
All right. The Morgan extract also uses a weak acid extraction to mimic what the plant can see. How is this test similar or different than the Haney test?
Yeah, I haven't messed with the Morgan extract in a long time or looked at it, like since graduate school. And while that wasn't very long ago, that was a really, really long time ago. So I haven't done any comparisons on those or looked at any data really to be able to answer that intelligently.
All right, and this is the last question since we're coming up on time. Dr. White, it's, I'm quoting from the person, this is not me talking, but it's my understanding that spoon feeding mineral or ionic nutrients to the soil influences or produces feedback in the microbiome. Example, adding ionic nitrogen will reduce nitrogen fixing bacteria. My question is, do we see the same feedback via foliar application of mineral ionic nutrients, whether in the soil or as endophytes?
You know, that's a really good question. and I don't know the answer. A really good question that I don't know the answer to. You're absolutely right. What you said, we put nutrients on the soil to give it to the plant, and we affect the system that plants are using to get those nutrients from microbes. And that's especially clear in macronutrients like nitrogen. And we know what happens with nitrogen. We put nitrate on the soil, and other too much chemical nitrogen, and it shuts down nitrogen fixation in the microbes, and they simply don't fix. And so you see in the plants, you see reduced nitrogen fixation in the tissues when you give them nitrogen. That system they normally use to get nitrogen doesn't work. It shuts down. And you see it in the soils, if you put the nitrogen there. Similar thing happens with phosphorus. We have not really looked to see all of the ionic forms that affect, that you can put on plants, either foliar or on the roots, and have impact on what's happening. So we don't really know. With a few nutrients, there's some evidence of what's happening as they're shutting it down. With others, we just don't know. That's all I can say, really.
All right, excellent. Well, we are on time here. Thank you both for your time. Thank you to everyone who attended. We will do our best to email the rest of the answers to your questions. And if you have any other concerns, feel free to email myself or Jamie here on the webinar, and we will see you next time.
Thank you.
Thank you. Bye-bye.
Thank you both. Have a great afternoon.